Hello, good morning. Our project name is Isolation and Identification of Alpha Amylase Producing Bacillus from Pulse Industry Waste and Estimation of Alpha Amylase. Our project guide is BB Sangameshwaran. Our group consists of D. Shrutakirti, Anil DK, Ravikiran T and A. Sachin. Objective of our project is to isolate the strain bacillus from waste of pulse industry and production of amylase assay and partial purification of amylase. And the other objective is to study the effect of carbon and nitrogen sources on production of amylase and also to study the effects of pH and temperature on amylase activity. Genus Bacillus is a gram-positive rod-shaped bacteria. Bacillus species can be obligate aerobes or facultative anaerobes. Test positive for enzyme catalase. Bacillus includes both free-living and pathogenic species. Amylases are the important enzymes, particularly in the process involving starch hydrolysis. They originate from different sources like plants, animals, and microbes, but they are mainly produced from microbes due to their higher yield and thermostability. Bacillus produce alpha amylase, which is a type of amylase. To isolate bacillus, pulse industry waste taken as source and isolation is done by serial dilution. Serial dilution is the stepwise dilution of a substance in a solution. Usually the dilution factor at each step is constant, resulting in a geometric progression of concentration in a logarithmic fashion. A tenfold serial dilution could be 1 molar, 0.1 molar or 0.01 molar, etc. Isolation is followed by identification of amylase producing bacteria. In this we study cultural characters like colony morphology, microscopic characters like spore staining, gram staining motility, and various biochemical tests are conducted and confirmed as amylase producing bacillus. After identifying the amylase producing bacillus, amylase production was carried out by submerged fermentation in favorable conditions. After incubation, fermented broth was centrifuged at 10,000 rpm for 5 minutes in a cooling centrifuge. Supernatant was collected and used for the estimation of amylase. Effect of different carbon sources on amylase production was studied using different carbon sources like fructose, glucose, glycerol, lactose, maltose, starch and sucrose, and it is found out to be maltose gives the maximum results. Similarly, effect of different nitrogen sources on amylase production was studied using various nitrogen sources like peptone, yeast extract, tryptone, soybean meal, beef extract, gelatin and red gram flour. Partial purification of amylase is done by ammonium sulfate precipitation method and estimation of enzyme activity is done by Barford method. Effect of pH on amylase activity is studied for different pH values ranging from 2 to 12. Effect of temperature on amylase activity studied for different temperature values ranging from 5 to 85. Major applications of alpha amylase include food, pharmaceutical and fine chemical industries, and it is one of the most important industrial enzyme known and is of great significance, having 25% of enzyme market. Our project study reports biological production of amylase bacteria bacillus species isolated from pulse crop processing industry waste. Enzyme production was found to be maximum in the presence of maltose as carbon source, peptone and yeast extract as nitrogen source at temperature 65 degrees and pH 6. Thank you.